So the last talk of this session is from Dr. Li Tao, and the title of his talk is Characterization of Molecular Isoforms in Protein Therapeutics by Electrophoresis, Liquid Chromatography, and Mass Spectrometry. Thank you, Arinda.
First, I would like to thank Pittcon organizing committee for giving us the opportunity to share with you our perspective and strategies in the characterization of biologics. I would like to thank the audience for staying here so late. So I will try to do my best and provide some useful information to make you feel by the end of the talk your time spent here is worth it. As you can tell, we have a pretty diverse group. Majority of the people here listed are mass spec person. Most of our work, since majority of our work use mass spectrometry, but we do provide chromatography analysis. As you can tell, the real power of mass spec actually comes from 50%, maybe from the separation. The rest came from mass spectrometry. By the way, our group is hiring biophysical people. If you have a biophysical characterization background, you can go to the BMS website, and there are a few positions in our group. So for today's talk, I'm going to touch upon the specifics of biologics, followed by a more detailed discussion of molecular variance characterization. In the end of my talk, I will going to show some of those kind of a new trend. The landscape of characterization, the focus changed over time, but more recently, sequence variance is one of those hot topics. After listening to you guys talk, I realized this, actually I dragged this from another slide and put here, just to provide a link between method development, which you guys did a wonderful job on, and our work, which is characterization. Just to put it simply, the characterization is about characterize the molecule, or how, just like person, how you look like, right? And then over time, everybody will age. Some of us may age faster under stress conditions, and some of those go slower. So we need to provide that information to method development to let them find the appropriate method and the parameters to monitor. And in the meantime, we need to have a long-term idea of how the molecule will come after storage, for example, or after manufacturing process. Then different groups, for example, method development process people can develop their method or modify their process and then keep the consistency of their drug product quality. I would like to mention here that protein therapeutics, which are the protein drugs, okay, are oftentimes called biologics. In reality, biologics also include vaccines, since the majority of the product on the market are protein therapeutics. So people use biologics, protein therapeutics interchangeably. The reason that biologics is gaining momentum in recent years is because, first, higher specificity, that is the result of a molecular interaction between protein molecules. Secondly, compared to the small molecules, it has a less toxicity, which translates into a higher successful rate during the process development and also clinical development. Differently than small molecule is the patent protection. So small molecule drugs, after you synthesize with a limited amount of analytical method analysis, you know exactly what it is. But for a large molecule, it is different. So where it is policy, as Hans mentioned, there's a lot of different things between health authorities around the world and the industry. So that area is still kind of murky, but it will evolve over time. I'm going to show you the biosimilar opportunities there, which Hans already talked about. However, in addition to the advantages, we do have disadvantage of large molecules, namely the complex nature of large molecules, and also the manufacturing process is much more complicated than small molecules. This is a figure that illustrates pretty much the so-called patent cliff, meaning after your drug is off patent, other people can make synthetic drugs, biosimilars, right? In the case of small molecules, after usually generic drug is launched, the first year or so, the sales drop about 80%. But for large molecule, it is different. Regardless, if you look at the next few years, the big sellers of biological drugs on the market right now, majority of them will go off patent. So by 2019 or so, the drugs with a combined market value of about 57 billion will be off patent. That's what Hans is talking about. There are a lot of opportunities over there for big and large and small companies to capture the opportunity, and also for young scientists looking for a career to get into. My personal feeling is biological drug is a good area for you to get into. The manufacturing process of large molecules almost invariably involves cell line development. You need to have robust, high-yield host cell to express the proteins. And then you have to have well-controlled process to let the cells secrete molecules consistently with high yield again. And then the downstream process will figure out the best way to purify your product with consistent quality and high yield too. So the yield is critical part among all of these steps. Since they already talked about it, I'm just going to skip it. But I just want to make sure that Dell and I guess we use different. Probably, I think the idea came from one person years ago, but I didn't know where it came from. It came from some colleague in BMS, but we did not copy it from Genentech, by the way. So how different is the large molecule characterization compared to small molecule? It is quite different. For example, only the primary sequence and the post-translational modification can be extensively characterized, and that's about it. However, it makes things more complicated. The protein function depends on not only the primary sequence, but also secondary, tertiary, and quaternary structures to make it fully active. That's why Hans showed so many orthogonal methods to guarantee or to make sure the generic drugs are similar to the innovative drugs. Traditionally, by the way, biologics have been controlled or regulated by the process, not the product. The reason is you cannot identify, characterize, analyze all those components since there are so many variable components in your complex. And ultimately, the safety and efficacy of your drugs will be tested in the clinical studies. So the first point I would like to make over here is the essence of characterization of biologics actually is on the isoforms. If I have a major form, it is much easier to characterize and look at its biological functions. The key part of biological characterization actually is on those minor forms which you cannot isolate, and then to perform an additional analysis. So where are those molecular variants come from? Well, if you look at the primary structure, we have variations, right? You guys already talked about it a few times. Glycosylation is one form of post-translational modification. In addition to that, the molecule will change over time. So deamidation, oxidation, isomerization, for example, are the major degradation pathways you need to pay attention to. In addition to that, fermentation is another thing that is almost always decrease molecules activity, right? And disulfide reshuffling, misfolded molecules, high molecular weight, low molecular weight species are all these parameters we need to pay attention to. As Dale showed earlier, the number of variants in complex usually, and his calculation is even much higher, but usually, depending on what kind of a platform, like domain antibodies, which has much less numbers of variants, to a large molecules like antibody, you have around a few dozen to millions, multiple millions sometimes. So what I'm going to do with these complex molecules, not majority of them we cannot even isolate. Well, you need to use cutting-edge technologies. So differently in the characterization work compared to Rendon's work for them, to make sure the method is well developed and make sure not only yourself, very talented people, but QC lab can perform the same study consistently. So you guys have more limitation in terms of work techniques you can use. In our area, you can almost use everything as long as a robust, reliable technique, okay? And then in our area, you can tell the different style of scientists working on the same thing, they may have slightly different results, but that is okay. In most of our situations, the reasonable quantification accuracy is less as stringent as in your case. So you need to have a cutting-edge technology and also take advantage of platform techniques. For example, capillary image capillary isolatory focusing nowadays is almost the norm. About 10 years ago, really not many people are using it. And Genetic is one of those companies that really push forward and use capillary-based techniques. And those are pretty much everybody's using it. In addition to that, some other techniques like HT exchange, MS, Hans already talked about it. We already use it regularly in our compatibility studies. And those are the things that we really pay a lot of attention to. In addition to that, probably the crown jewel of all analytical testing method is the so-called cell-based bioassay, so which has to rely on live mechanism, live cells to show the mechanism of action of your drug. So that is much harder to develop, validate, but however, that is the most important analytical techniques to guarantee the consistency of your drug product. Another thing we could do is to take advantage of well-developed platform. For example, monoclonal antibodies. Actually, at the BMS, we try different platforms like domain antibodies and nectins, but monoclonal antibodies being one of the largest natural molecules, somehow delivered better than other platforms. So it is not surprising. Majority of the commercialized products are based on the monoclonal antibodies. The thing we like monoclonal antibodies is it has a very well-known glycosylation profiles and C-terminal heterogeneity, ionization, oxidation, all these kind of things. So not only you make yourself comfortable, for health authorities, if you have a well-developed product that is based on a well-known platform, it makes their work much easier to guarantee the safety, or actually more importantly, efficacy of your drug. Talking about fermentation, I think this is pretty consistent among all different monoclonal antibodies. I think the work was published 9 years ago. Based on our experience, it is quite true, which is fermentation of monoclonal antibodies almost always happens near the hinge region. Of course, sometimes it happens on other regions too, but the majority happens to the hinge region. This map pretty much illustrates the predominant fermentation site, which are shown by these darker squares and those minor ones, right? So what if your molecule is different than monoclonal antibodies? Or you do have such cleavage site that is not in the hinge region. Nowadays, mass spectrometry can pretty much tell the answer quite easily. So being a mass spec person, maybe we are more biased towards mass spectrometry, but compared to all other techniques, there's no other technique really can compete in terms of the content of information that is generated, okay? So in this case, nowadays, if you have a known knowledge of the sequence of the molecule, and you have some fragment, just by simple measurement, and then with some kind of assistance of the patent matching, you can pretty much figure out where it started, where it's at sequence-wise. There are two papers talking about this. One is from us, which is highlight one of those really quick, accurate turnaround of this technique. Another example is the glycosylation. By simple mass spec analysis, even without the need to digest into peptide, you can already tell the glycosylation profile of different glycoforms. Again, as I mentioned, you need to rely on cutting-edge technologies. And this is a result obtained on thermal Q-exactive. You can tell the difference between this and the older Q-TOFs in terms of resolution and accuracy. And most of the time, Dale mentioned he doesn't like peptide mapping, but that's our bread and butter. Everything we cut into peptides, almost everything. And not because we like it, but that's the only way we can do it. The power of peptide mapping with LC-MS comes from the highly efficient separation by reverse phase. A small difference in the peptide, and as long as the peptide is not too big, can almost always be separated. That's really the power. With this figure, which shows the different glycosylation pattern from different lots, that is something we need to figure out when we do comparability studies, but it's not always easy. And you do see differences. How much of difference contribute to the end result is a totality of the data. And different assays provide different weight in your final determination. In terms of deamidation isomerization analysis, peptide mapping, of course, is not as straightforward as CEX method as Dale showed earlier, but probably it is most accurate because it can pinpoint to which deamidation site and how much of each site is deamidated or isomerized. As I mentioned earlier, if you deal with antibody molecules, significant amount of CDR region, your isomerization almost always comes from CDR region because FC part does not have too much isomerization. It deamidates faster most of the time than CDR region, but isomerization is very little. Another point I would like to make is method-induced deamidation. The protein molecule has a more constraint from conformation, so the flexibility between amino acid residues is more constrained than after you cut into peptide. So almost always after you cut into peptide, the deamidation went much faster. So you need to be careful how much is really method-induced, meaning after you cleave into peptide, compared to before you cut into peptide, how much is there already. To make it really, really accurate, you can go crazy to do it in the way of digesting your peptide in an isotope-labeled solution, and then you can differentiate by mass spec how much is pre-existing, how much is method-induced. For oxidation, same thing here. Mass spec can give you a pretty good answer by looking at, again, since we have knowledge of this protein molecule already, methionine residues are almost always the most susceptible oxidation site. We can tell how much is there and how much is on each site is oxidized. And again, majority of them actually are not in the CDR region. And we would also like to mention here that the oxidation usually is not a major degradation pathway. So you don't have to spend a lot of your resources developing a method to monitor oxidation. But in order to achieve that conclusion, you need to have data from forced degradation study, where you force the molecule to oxidize. And then you need to have certain stability, looking at the time point of your stability sample to make sure oxidation really did not increase too much. And also, you need to correlate oxidation levels from different sites with your biological activities. And then with this information, you can just skip method development. I'm not saying method development is not important, but just based on the pain you guys showed us, if you can avoid one method to monitor release and stability, you really save a lot of resources and money. But another point I'd like to make is mass spectrometry cannot do everything, right? Just to want to give you an example. In the case of charge variance characterization, mass spec can do some things, but for accurate determination of charge variance, we'd better leave it for CEX, ICE, that kind of method. The reason, actually this is another hot topic in recent years, the reason that charge isoforms are important for the characterization of biologic molecule is because almost majority of those degradation pathways involve a change of a charge on the molecule. And also the performance and stability of your process usually has something to do with the charge profile. So by monitoring the profile of a molecule, you can make sure your process is consistent. Your molecule did not change too much. Another reason that everybody has to put a lot of effort to do this kind of work is health authorities. They knew it. Among all those different isoforms, charge isoforms are easiest to isolate and characterize. They're hydrophobic variants. There are like conformational difference. Those are much difficult to deal with. So for charge isoform characterization, you have orthogonal techniques, which is important for the biological drug characterization. For peptide mapping, it can be used almost everywhere, but for quick turnaround and more accurate measurement, ICE is a preferred method. If you want to isolate charge isoforms and then perform additional study, ion exchange, Hick, those are more powerful. So we have to make sure, while we realize we want to use peptide mapping method all the time to provide comprehensive information, we do not want to develop a peptide mapping method. I fully agree with Adele. Peptide mapping method is much easier to perform as a regular stability indicating method. The reason is that you have to put the enzyme. The enzyme has to cut your protein. It has to cut consistently, right? You have so many variables. And then sometimes peptide mapping pick up unnecessary details, isoforms, that takes a lot of time to explain. And it may come from different sources due to the quality of your enzyme, due to the reaction time variation, that kind of thing. So ideally, if you have any other method, such as ICE, CEX, that use a chromatography technique to quantify directly, you do not want to cut into peptide and do this kind of thing. Sorry, I actually already mentioned this. Another example, that peptide mapping lose to IEX and ICE method is its sensitivity, not only an individual site, but on the overall molecule. For example, deamidation happens on multiple sites. But when you look into these individual amino acids, the level is very small. You cannot pick up easily, which is illustrated by this calculation here. Let's assume there are 10 residues that can deamidate. Only if the individual level of deamidation is 1%, okay, the total amount of deamidation is 10% over here. That can be picked up pretty much easily by chromatography techniques. But by peptide mapping, you look at individuals at 1%, it's almost impossible to do it. If you have a more higher level, for example, 2%, that is very obvious. But 2% of change by plasma mapping is still very difficult. Okay, if you have more than 10 residues, which is the case for monoclonal antibodies, and then the difference is huge. We have a real example over here. This is a map going through a force stress degradation condition and 45 degrees for 6 weeks. 6 weeks and 45 degrees is pretty harsh. The ice electrophoregram showed quite obvious change, whereas peptide mapping, looking at the most susceptible so-called deamidation marker, still has almost not much change at all. Right. So if you have an overall deamidation method, you certainly do not want to use peptide mapping. Okay. The combination of everything combined together can be measured without the digesting. That's the way to go. Last of my talk, I would like to talk about sequence variants in biologics, which is the area that more and more companies pay attention to, and the health authority also realized this is something we may have missed earlier due to the limitation of earlier generation instrumentation. So sequence variants comes from so-called amino acid misincorporation, meaning there's a mutation somewhere, point mutation of a one amino acid being different, which is a result of several stages of reaction. For example, during DNA replication, if you have a mutation, that can result in a new amino acid sequence. Or it can be during the DNA to RNA transcription, right? And it can also happen during protein translation. Everything is okay, just like human beings. Nature may make mistakes over time. Everything is okay. And the last step, when the tRNA carries amino acid to the polypeptide chain, it put in the wrong stuff there. The chance is different depending on different stage. For DNA replication, the chance is pretty low. But when you come to the protein translation, on the different conditions, that can happen almost 1 in 1,000 kind of probability. Other factors also contribute to the amino acid misincorporation, such as cell-selecting reagent, where you develop your cell line, and also if you have reactive oxygen species in the formulation, or, in the expression media, you may introduce amino acid misincorporation. Other factors like amino acid starvation. If you have a media, expression media, and that does not have enough serine, and sometimes the cell will bring asparagine to that site because these two look very similar. So actually, in terms of making a robust cell line, I think the easiest way to do is probably codon optimization, meaning certain codons, like AGA, tends to have this arginine to lysine mutation. And same thing here. If you have an AGC codon, it may have a serine to asparagine. It may contribute to a higher level of asparagine, serine to asparagine misincorporation. So is amino acid misincorporation preventable? Well, depending on which stage we are talking about, during the DNA replication, it is really not much you can do. During mRNA transcription and the protein translation, you can do some codon optimization. As I mentioned earlier, additional measures such as slowing down expression rate may help a little bit. If you make the cell work too hard, cranking out proteins too fast, the quality may suffer. So very similar to our human workforces. From evolution's perspective, actually a certain amount of ambiguity in terms of what kind of codon transfer to what kind of amino acid to incorporate into the polypeptide chain is a natural. There's some natural significance. If I have absolutely same codon transferring to the same protein sequence, probably you won't have very robust evolution significance. Meaning, if everything is the same, there's no change, there's no adaptation to environmental pressure. So don't be too afraid of seeing this. Actually, if you go back looking at the protein molecules, I'm pretty sure you're going to see a lot of low levels of so-called common misincorporation. For example, we found out serine and asparagine got replaced each other quite a lot, especially if you go to lower levels. If you go to the 10 to the minus 4 or minus 5, this kind of thing can happen all the time almost. So it is surprising. With these newer advanced mass spectrometers, this thing can be picked up already. But however, you do not have to be alerted. However, one thing we need to make sure is your cell line is homogeneous. If your cell line, which you use to express your protein, is not even homogeneous, for example, you have two cell lines. One population expresses one protein, which is slightly different than the other cell line, then depending on which stage are you in, you could have really serious problem because you cannot control the population being exactly the same ratio over time. So what are the challenges to determine the amino acid misincorporation? The real challenges are, most of the time, you need to analyze a sample, right, which consists of majority, overwhelmingly correct sequence, but hidden in that population, there are a few percentage, sometimes even less, of amino acid misincorporated sequence. And that is the real challenge. Most of the time, this happens in the cell line development stage. You need to tell the cell line people, this is homogeneous, or this is not homogeneous. It is not always that easy. So, but we do have certain tricks to provide critical information for cell line development. For example, oftentimes we think less sample, right? It's easier to have a quicker turnaround if you have fewer samples. But in this case, it's different. If you have a clone that comes, if you have samples, right, they come from different independent clones. The chance of all these clones having the same amino acid sequence on that same site, the chance is very low because it's random. So by combining them all together and random, you can quickly rule out. If you don't have to look at this larger peak. If there's a small peak that happens from all independent clones, most likely it's not sequence variance. It is something from your enzyme, from missed cleavage or over cleavage product, or specifically cleavage product, that kind of thing. That can easily focus on something that, in this case, this is a real example of sequence variance. It's a few percentage, but we were able to pick it up quite easily by doing this. And that can significantly improve your efficiency, especially if you have multiple samples. Some of those are from independent clone, some are not. And then just by combining these patterns, and then you can turn result much faster. On the other hand, if you have only one sample, it is much harder to do. So the current situation is for cell line development, the key is to make sure there's not or very little or non-detectable sequence variance that is due to the heterogeneous cell line. So we have to make sure we have a very high true positive rate, right? But life is a dilemma. If you have very high positive rate, you're going to have higher negative rate, right? If you don't miss any amino acid misincorporation, you're going to have a lot of those that looks like they are, actually they are not. And that's technically the bottleneck in our work. We use mass spec and search all possible any amino acid can be replaced by another one. By point mutation, we search against it, right? And then you have large number of so-called false negatives. And then in order to rule out those are not from the heterogeneous cell line, takes a long time. So that's where technically bottleneck is. In summary, I would like to once again emphasize that the biologics are a mixture of multiple variants, okay? And then very few of those product variants can be individually characterized. You cannot separate them, isolate them, you forget about anything else. Also, mass spectrometry plays a central role in protein therapeutics characterization, but other orthogonal techniques such as electrophoresis and liquid chromatography are essential too. With that, I would like to acknowledge the support from upper management, Jay and Maury. I would like to entertain any questions if you have. Thanks for staying so late.
Do you have an idea on the sum of sequence variants present in percentage? So the sum, not the single one, so summing all.
Depending on the level, there was a paper published by Zhongqi from Amgen a few years ago. If you look at 10 to the minus 4, minus 5, I think almost all proteins have a certain amount of sequence variants there. Because the error rate is 10 to the minus 3 and 4. You have a long peptide chain, mistakes will be made.
Any other questions? If not, thank you very much.
Thank you.
That finishes our session. Thank you again.
You don't need to do anything this way.
Yeah, I'm just going to delete everything. Sure.